All right, well, hello everyone. So we're going to look here at our kidneys, our renal system, and in particular, what we're going to begin looking at in this part, in this kind of next set of videos, is one aspect of renal physiology called glomerular filtration, right? So there's going to be a sequence of events that we're going to talk about and I'll get to them in just a little bit. However, let me preface all of this by saying that in order to understand these next sets of videos that we're going to do, looking at something called glomerular filtration, we're going to look at something called uh, tubular reabsorption, tubular secretion. All of these are processes involved in making urine. In order to understand these, you have to know the anatomy of the kidney. Okay, you have to know the microscopic anatomy of the kidney, especially the nephron, right? So if you have not viewed the videos, if you have not looked at the anatomy of the kidney, and again, especially the nephron, that small microscopic functional unit of the kidney, make sure you do so before you watch any of these physiology related videos. Okay, so in this first part, what we're going to look at is we're going to look at one of these three phases here. And in particular, what we're going to look at is something called glomerular filtration, right? So that this little box that you see to the far left, right? Glomerular filtration, the movement of substances from the blood within the glomerulus into the capsular space, right? So this is going to involve the blood that we have circulating in the body. It's going to flow into the kidney, right? And remember, recall the flow of blood into the kidney, renal artery, and then we begin branching segmental. We get smaller and smaller until we come into the glomerulus through something called the afferent arteriole, right? The way you remember that is afferent, think A, arrive, right? The blood arrives at the glomerulus, and then it leaves via this efferent arteriole, think E for efferent, think exit, right? So we're coming into this glomerulus, and remember, the glomerulus, this is just a ball of capillaries, right? It's just a small, tangled ball of capillaries. And then within this glomerulus, there's going to be an interaction among different pressures, right? And we'll get to the pressures here in just a little while. But these pressures are essentially the same types of pressures that we looked at in previous videos when we talked about, the movement of materials in and out of the capillaries at the level of our systemic tissues, right? So whenever we had blood leaving the heart, left ventricle, aorta goes all the way down to our capillaries and our tissues, and you had the exchange of nutrients, right? That capillary exchange, right? It's going to be basically those same uh, processes, those same pressures, right? Hydrostatic pressures, osmotic pressures that are acting here in the kidney as well, right? And then we will have separate parts and separate videos on tubular reabsorption and tubular secretion, right? So this glomerular filtration, this first part that we're going to focus on, and kind of in these next several sets of videos, this has to occur first, right? You have to have filtration ultimately before you have reabsorption, and then secretion is kind of something slightly different, right? And we'll get to that in just a little bit, right? But we're going to look at lots of things that are filtered, okay? So essentially think of the material that's entering this Bowman's capsule, right? Or that space deep to Bowman's capsule, right? Or that capsular space. These are things like water, small solutes, glucose, vitamins, things like that. Larger things, so think red blood cells, white blood cells, large proteins. All of these are not filtered. Okay, they're too large. So um, you can think of this in the context of something called a urinalysis, right? And a urinalysis is essentially an analysis of urine and the components of urine. And there should never be blood in urine, right? There should be no red blood cells that pass this filtration membrane that we'll talk about and end up in the what's called filtrate, which is it's going to become urine, right? We'll then talk about reabsorption, right? Reabsorption, this is not the same thing as absorption, okay? Absorption is when you absorb something for the first time, and reabsorption is when you do it for the second, third, fourth, fifth times, and so on, right? And then secretion is moving things from the blood into the tubular fluid, and the tubular fluid is the fluid within these proximal distal convoluted tubules, these loops of Henle, and so on. right? So for these different processes, I want you to think about, and again, we'll go into more detail on each of them, but I want you to think about the direction that things are moving, right? So are we moving from the blood into the capsular space? Are we moving from the blood into the tubules? Are we moving from the tubules back into the blood, right? Which direction are things moving? And by things, I mean the water, the solutes, all the things that were filtered. Okay. So what you need to consider when talking about this process of filtration, reabsorption, secretion, is how these interact to produce urine, right? So we're going to use several different terms. We're going to use terms like um, capsular fluid. We're going to use a term called tubular fluid, filtrate, urine, right? Urine is the final product. Okay, so urine is what you excrete out, right? Um, Filtrate is um, the fluid that you produce after you undergo glomerular filtration. And then you've got your tubular fluid, which is going to be altered through tubular reabsorption and tubular secretion, right? So essentially think of like the filtrate, think of the tubular fluid as pre-urine, okay? It's going to become urine, but it's not final urine yet, right? Once you leave the uh, collecting duct, essentially, which again is kind of associated with our nephron. Once you leave the collecting duct, it's essentially urine at that point. There's no more modifications that are going to really occur, right? So when you're talking about the amount that we're going to excrete, the amount of urine, you have to take the amount that you filter. So that's promoting the formation of urine, minus the amount that you reabsorb, right? That material is going back into the blood, so it's decreasing the amount that you excrete, plus the amount that you secrete, right? So these terms here, secrete and excrete, are different, right? So excrete is, think like leaving the body. Okay, so you excrete urine, it completely, totally leaves the body. Secretion, right? This word here, secrete. This is moving the material, the substances inside the body from one area to another, right? So, in other words, you're moving things from the blood into the tubules. Okay, so it's not leaving the body yet. You're just moving it to different places within the body, right? So keep that in mind, right? So you can look at some of these animations and directions here, right? So filtration, we're moving from the blood to the tubular, capsular fluid. Reabsorption, we're going from this tubular fluid back to the blood, and then secretion is the exact opposite of reabsorption. So we're going from the blood into the tubular fluid, right? And then we'll continue on from there, right? So if we look, and again, we're going to focus primarily in these sets of videos on filtration, right? So for filtration, our water, our various solutes, all the things that we're going to filter out, they have to pass through different layers. Okay, they have to pass through different layers to ultimately leave the blood and then end up in the Bowman's capsule, that capsular space, right? So first thing is we're in the blood, right? We're in that glomerulus. So you have to pass the capillary, wall that lines the glomerulus. And so that's an endothelium, right? So that's a simple squamous epithelium. In particular, it's a fenestrated epithelium, right? Those capillary cells are fenestrated, meaning they have little openings, right? They have these small gaps in them that allow certain sized molecules to leave, but they restrict the movement of larger materials. So again, proteins, red blood cells, white blood cells, things like that, right? Deep to the endothelium, you have the basement membrane. So again, think essentially what the endothelium is sitting on, right? What kind of anchors it, what it serves as its foundation, its basement, right? Is the basement membrane. And we'll look at pictures of these. And then you have something called the visceral layer of this glomerular capsule, right? So you've got a kind of a parietal layer that we'll talk about, and then you have this visceral layer. Remember, viscera means on the surface of something, right? So it's this structure that's on this outer layer of our capillaries, right? So think a capillary sitting on top of the capillary. You have this visceral layer, right? So one thing that you have are these cells called podocytes, right? Podocytes. So if you take this word podocyte, podo means foot or refers to foot, and then cyte means cell. So these are these cells that have these little foot processes, and these foot processes are called pedicles, right? Pedicles. Pedicles are these extensions of the cell that, when they come next to each other, they create this space, and this space is called a filtration slit, right? So what you can see here are, here's a podocyte, this large cell. Here's our capillary, right? Notice the fenestrations, right? Notice those little holes, if you will, in the capillary, right? Those are the fenestrations. Here's your podocyte. Here are the pedicles, right? Those long kind of extensions, right, coming out. And then in between those adjacent pedicles, you have a little gap, right? And those are your filtration slits, right? So essentially, think if you hold your hands up and you kind of interlock your fingers, there are, not interlock your fingers, but you kind of hold your fingers next to each other where there's a space in between adjacent fingers. You've got these little openings, and those are your filtration slits, right? And so depending on the size of the filtration slit, you're going to be able to allow certain sized molecules to pass through if they're small enough. And if they're too large, then they're going to be restricted, right? And so what you can see in this graphic is you've got your various layers, right? So here you've got your capillary lumen. Okay, so this is the inside of the glomerulus, right? So you've got your blood, you've got everything that's in the blood, right, in your capillary, right? So formed elements, plasma, solutes, proteins, everything, right? Here's your endothelium. Notice the gaps, right? Notice your fenestrations. Here is your basement membrane that the capillary is sitting on, right? And then here are your uh, pedicles with your filtration slits, right? And so depending on the size of the molecule, right, you're going to be able to pass through, if you're a certain sized molecule, you're going to be able to pass through the fenestrations, through the basement membrane, and in between the pedicles through the filtration slit, right? So again, filtrate includes water, glucose, amino acids, ions, urea, hormones, vitamins, small amounts of protein, right? Um, Now, not lots of protein, but very, very small amounts of protein. And then honestly, we end up reabsorbing a lot of that, right? And so as we'll see, we create far more filtrate than what we actually uh, excrete in our urine, right? So filtrate, we produce somewhere around 180 liters of filtrate a day. Okay, so think 180 liters a day. Do you excrete 180 liters a day, right? Do you urinate 180 liters each and every day? No. Okay, not even close. All right. So what does that mean for the rest of the fluid, right? So obviously we have blood flowing through. Okay, we have blood flowing through the glomerulus. It gets filtered out. Some of it gets secreted. Some of the things get secreted into the tubular fluid. But then most of it we just reabsorb, right? So it gets taken back into the blood. It circulates, and then it'll circulate through the body. It'll come back to the kidney to get filtered again, and so on, right? But um, we are not excreting the majority of what we filter. Okay, most of it ends up getting reabsorbed, right? So this filtrate, again, that what is ultimately being collected within our that glomerular capsule, right? That Bowman's capsule, the capsular space. It's plasma, some amounts of solutes, and small amounts of protein, right? From that capsular space, it is funneled into the proximal convoluted tubule. From there, it goes to the descending loop of Henle, ascending loop of Henle, into the distal convoluted tubule, collecting duct, and then ultimately into our ureter, bladder, urethra, and excreted out, right? Um, so again, big thing here, the filtrate, again, certain things are not filtered because they are too large, right? They are trapped within the basement membrane. They are retained within the um, actual blood, right? You also have these cells called mesangial cells, and these are kind of around our capillaries. They support the capillaries, but they can also change the size of the filtration slits as well to alter the surface area to allow certain amounts of things to be filtered. Or potentially not be filtered, right? So these are different things that are, or different categories of things that can be filtered, right? So things that are freely filtered, things that are not filtered at all, and then things that are maybe somewhat filtered, right? Limited filtration. So freely filtered, these are small, small things, right? So water, glucose, amino acids, ions, right? Really, really small substances. They go through the fenestrations, they pass through the basement membrane, they go through the filtration slits, no problem, right? Anything that is much larger, large proteins, any of our formed elements, red blood cells, white blood cells, those are not being filtered. And then limited filtration, think essentially kind of uh, medium-sized proteins that could potentially pass through, usually not, um, but they could, right? So due to their size or due to their electrical charge. Okay, so this is where we're going to stop for this part. And then we will pick up in the next part looking at some of those pressures that I alluded to, some of the pressures that actually influence the process of filtration.